Okay, good morning, everybody. Hope everyone is doing okay with the last part of the term upon us. So you'll be happy to know that this is the last lab for this term for Geography 270. So we're going to be continuing on in spatial analysis. So this is the second part of that, in which we're going to be using again the application of Geog to do our spatial analysis. So again, we're going to be looking at data collected for the country of Nepal, although the data file is a different download. So you'll see it in the Lab 8 Moodle folder there. It's called Nepal 2016. And again, you'll need to download it and unzip it. And when you do, you'll find that there's various different types of files contained within that package. So everything from Excel files to CSV files, geo package files, Excel files. Um, but pretty much GeoDoc can use all of those, but the ones that you're probably going to want to use in GeoDoc are going to be either the shapefile or the JSON files. Those are the main ones that should work to use in GeoDoc to complete this lab. So again, we're going to be doing similar things to last time. We're going to be looking at doing a Moran's eye test, but now we're going to be also looking at doing a local Moran's eye to see where we have hotspots of clustering or non-clustered data. So other types of tests that we looked at in the lecture, GetHG, and other clustering algorithms in GeoDA. So again, we're going to be looking at the Nepal data in the 30-some districts in which data is collected for a whole bunch of different variables. But the variables we're going to be looking at in today's lab, two main ones. One's coded kids, which is the percentage of children under 5 who are malnourished. And then the other variable is PC Inc., which stands for per capita income. So those are the two main variables we're going to be analyzing using GeoDA in this lab. Okay, so the first thing, as always, we're going to visualize our data. And in GeoDA, there's various ways we can visualize our data. A common way is to visualize our data using what's known as a cartogram. Basically, a cartogram represents our data in a couple of different ways visually, by using different colors and also changing the size of the circles. So basically, you're taking the map of Nepal with all the different zones, and you're transferring those into either different size circles and different colored circles. So the first thing we're going to be doing is making a cartogram using the variable malkids as both the circle and the color in the cartogram map. And then the second cartogram you're going to make is going to be looking at malkids, and you're going to be describing malkids in terms of the circle size. And then the circle color is going to be set to PC Inc., per capita income. So once you do that, you can save it and then download it or upload it into the Dropbox. Right? So a couple of different ways you can do a cartogram. There's the icon, and then it's also underneath the map tab in GeoGebra. So here's an example of a cartogram using different data. So you can see again, right, how this single variable is quantified or visualized both in the size of the color or the size of a circle, sorry, and the color of the circle itself. So you can see it goes from low values, which are small and blue circles, up to high values, which are big and red circles. So that's essentially what a cartogram does. Okay, question 2, looking at the cartogram and using the what's known as the Hinge 1.5 classification, which is the default setting in GeoDA, so you shouldn't have to change that. Looking at your two cartograms, right, which of the two cartograms show upper outliers, right? So there's your two choices there. And then what do the upper outliers in the data set mean? Okay, so 3 options there. So, right, there's the hinge set to 1.5 there for this example for the cartogram there. You can see where the upper outliers is set. Second thing we're going to do to visualize our data is create a spatial correlogram. So a correlogram basically shows us how much our variable relates to itself in terms of correlation over space. So we're going to be looking at whether mal-kids variable varies over space, and if so, how does it vary? So under the space tab, you're going to be choosing the spatial correlogram, right? And you're going to be setting the settings for the correlogram as you can see on the tab there, right? So you're going to be looking at the mal-kids variable. You're going to set your distance to arc distance, number of bins 10, and then click the radio button for all pairs. And this is an example of a correlogram right here. So there's two components to the correlogram, the bottom histogram, and then the top line graph shows us how the variable is autocorrelated at that distance. So right there, where we have the line above the 0 mark, that hash line right there, that tells us that at that point, over that distance, our variables, our values tend to be positively autocorrelated, aka clustered. In this example, we can see at far away distances, right? The Euclidean distance are along the x-axis there. So at very far away distances, our variable values are actually negatively autocorrelated. So it's actually more dispersed than you expect randomly. So that's how you can interpret one of these correlograms. So set the correlogram 30 bins for the histogram, and then based on that, answer in approximately what distance range does the spatial autocorrelation for the variable mal-kids turn negative. So you should see that that line that I just showed you at some distance goes from being above the 0 line to below the 0 line. So you're just going to select at which distance that occurs. Second thing you're going to do with the correlogram is that you're going to right click on the trend line, that upper trend line, and you're going to change the bandwidth from 0.2 to 1, and then basically indicate from those 4 choices what that does to the trend line. Okay, a little bit more of visualization. We're going to create a scatter plot where our independent variable x is going to be mal-kids, and our dependent variable y is going to be PC per capita income. So you're going to be creating that scatter plot and determining what is the coefficient of determination, the r squared, for those 75 observations. So there's the icon for a scatter plot right there. So once you get your scatter plot created, you'll notice that you can hover over top of it, and you can select certain points or certain data points within the scatter plot itself. So you'll notice that there's 2 outliers above the 1300 level of income in terms of per capita income, right? So if you scroll down, you can see the summary stats below it, the scattergram, right? What is the r squared when you eliminate the 2 outliers? Again, you can do this by just dragging over top of the scatter plot itself. Okay, so you'll probably see that the scatter plot does not seem to indicate a big, strong correlation between per capita income and malnutrition in kids. So what we're going to do for some further analysis is we're going to do a Moran's I. And so to do this, we're going to have to create a weights file. So we're going to be creating a queen contiguity file. And once you do that, you're going to go down below and you're going to create what's known as a connectivity graph, which is a visualization of how the polygons or the zones are connected to one another in your data. So here's an example of a connectivity graph, right? So it just shows you the connection of the polygons defined by the nodes. So it gives you a visualization of the spatial relation between different locations. So once we got our weights file created, we're going to first do a univariate local Moran's I Lisa analysis, okay? So a univariate means that we're comparing the value of one variable, right, against itself, okay? So in this case, right, basically you're going to be making a significance map, a cluster map, and a scatter plot. So of course, this is going to be, as you can see up there, the variable in this case is going to be mal-kids. So you're going to be doing the univariate Lisa using the mal-kids variable. Create your significance map, cluster map, all those things, and answer below, what is the Moran's I numerical slope up to 3 decimal places? So there's an example of the Moran's I scatter plot right there. So there's the Moron's I. In this case, it'd be 0.282. And then also generate your significance maps, right? Different greens show you different levels of significance in clustering. And then the map on the right, the reds show you where you have high values against high values. And then the blue values show you where you have low values against low values. And also low versus high and high versus low. So also create those maps. Then once you're done that, you're going to run your permutations. You're going to run 999 permutations to generate your pseudo p-value, which you're going to be using to determine whether the autocorrelation, the Moron's I, is statistically significant or not. So you're going to be entering the p-value that you get from that to 3 decimal places in the space provided there. And then based on that p-value, are you going to reject or not reject the null hypothesis given an alpha level of 0.05? Okay, so that's the LISA, or the local Moron's I. We're going to compare the results of the LISA to another local clustering test called the local G star. So you're going to be running the local G star. You're going to click the button to generate a significance map, a cluster map, and then you're going to be using the results from both the LISA and the G star to basically indicate by dragging and dropping the labels at the bottom to the locations in the map itself. Okay, so now you're going to change the significance of your G star cluster map to 0.01. So a higher threshold, right? So this should actually reduce the number of areas covered. So now you should be able to see what areas are still significant once you've changed the significance level to 0.01. Click the locations, right? The zones listed below that are still significant. Okay, so once we've done our univariate local or LISA, now we're going to do a bivariate LISA. So in a bivariate, we're comparing how one variable at one location compares to another variable beside it. So for the first run, you're going to be doing a bivariate local Moron's I using per capita income for the X variable. And then the Y variable, the second variable, is going to be male kids. So again, you're going to be reporting first your Moron's I to 3 decimal places, and make sure you indicate whether it's positive, right, or negative by using a plus or a minus sign. And then based on the results of this first BiLISA or bivariate local Moron's I cluster map, fill in below the number of districts for each group type, right? So how many of the districts have high, high, low, 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 high, or high, low, right? So again, you're going to be looking at your significance map and the type of clustering that is generated in this bivariate LISA. So to do a little further analysis, we're going to be creating what's known as a scatter plot matrix to see if any other variables might be affecting the relationship between male kids and per capita income. So basically what you're going to be doing is choosing male kids, per capita income, and 2 other variables to make a scatter plot matrix. Okay, so essentially this is an example of a scatter plot matrix where you have your variables right along the bottom and the top, and it's a way to compare to see whether you have any correlation between 2 other variables. So just look at 2 other variables, see if there's any other correlation going on between those 2 variables. And then what you're going to do for the end of the lab is that you're going to run another bivariate LISA, but this time you're going to change the X variable and the Y variable. You're going to be basically switching them around. So in this case, you're going to be putting male kids as the X variable and per capita income as the Y variable to see how the results from this bivariate LISA compares to the first one. So once you compare the results for both the BiLISAs, right, analysis where you've switched around the X and the Y variables, what is the new, first thing you're going to do is what is the new Moron's I for the BiLISA where you switch the X and the Y around? And from that, explain whether the male kids and the per capita income lag variables have a statistically significant spatial relationship and the direction of that relationship, and whether it's strong, moderate, or weak. And the third thing, is this relationship stronger than the previous bivariate Lisa? Okay. And then basically at the bottom below gives you some idea of why there might be differences in the Moran's eye and the type of spatial autocorrelation you might see between the two variables by just switching them from the first variable to the second variable. And that's the end of our final lab. So in this lab, we have no final exam, although do keep in mind that some of the things that we've done in the lab likely will be on the final lecture exam. So don't forget it completely once you're done this week's lab. All right, as always, have a good weekend, and we'll have one more lecture that I'll be dropping for you guys later this week.